Your immune system exists to shield you. It tracks down viruses, bacteria, and other invaders with almost military efficiency. But what happens when that same system turns on you? What if your own defenses started attacking the very neurons responsible for keeping you awake? For decades, researchers wrestled with this question, and a landmark advance in genetics has finally delivered an answer that upends everything we thought we understood about one of neurology's strangest conditions. The story starts in 1877, when French neurologist Jean-Baptiste Edouard Gélineau first documented an unusual sleep condition patients who could abruptly drop into deep sleep at any moment with no warning at all. The disorder was so bizarre that for well over 100 years, physicians treated it as a psychological quirk, a character flaw, or simple laziness. Patients went from clinic to clinic looking for answers, and were instead told nothing was physically wrong with them. They were accused of lacking discipline, of being lazy, of suffering from depression or instability. The emotional weight of being disbelieved was almost as crushing as the symptoms themselves. Then came a turning point. In the 1960s, scientists identified a small group of neurons deep in the hypothalamus that produce a signaling molecule called orexin, also known as hypocretin. This chemical does something crucial. It sustains wakefulness, stabilizes the sleep-wake cycle, and helps regulate REM sleep. When these neurons work as they should, you stay alert and in control of your sleep patterns. But what happens when roughly 90 of these orexin-producing neurons simply vanish? For years, no one could explain why these specific cells were dying while neighboring neurons stayed untouched. The selectivity of the destruction suggested something was deliberately targeting them. Raising an unsettling question, was the body attacking itself? In 1997, researchers found that nearly all narcolepsy patients shared a specific genetic marker, a variant of the human leukocyte antigen HLA system, the mechanism the immune system uses to tell self from non-self. Almost 98 of patients carried this particular variant, an almost perfect correlation pointing straight at the immune system. But there was a puzzle. That same HLA variant appears in roughly 20 of the general European population. If it alone caused the disease, 1 in 5 people would have narcolepsy. Yet the condition affects only about 1 in 2,000. Something else had to be involved, some additional trigger, required to activate the danger hidden in that genetic variant. Scientists turned to environmental triggers, infections, vaccines, immune challenges, and found something striking. After the 2009 H1N1 flu pandemic, narcolepsy cases spiked sharply in certain populations, especially in Scandinavia. The timing was too precise to be coincidence. It suggested that a specific pathogen could set off narcolepsy in people who were already genetically vulnerable. The missing piece came from understanding cells, the immune system's roaming patrol units trained in the thymus to distinguish invaders from the body's own cells. Cells carry receptors that bind to foreign fragments presented by HLA molecules, like a key fitting a lock. When that fit occurs, the cell activates. These receptors are generated through a process called VDJ recombination, which shuffles genetic segments to create nearly limitless diversity, giving each cell a unique receptor tuned to a single target. This diversity is what makes the immune system so powerful, but it also means some cells could in theory recognize the body's own tissues. Normally, these self-reactive cells are eliminated during development in the thymus. When that filtering fails and dangerous cells escape into circulation, autoimmune disease can follow. Researchers began asking, were people with narcolepsy producing self-reactive cells because of their genetic makeup? Were genetic variants in the cell receptor gene letting these dangerous cells slip through? In 2009, genome-wide studies revealed something remarkable. People with narcolepsy also carried unusual variants in the gene that encodes part of the cell receptor TCRA. This was the second critical piece of the puzzle. The HLA variant created the vulnerability. The TCRA variant enabled the attack. People carrying both saw their risk of developing narcolepsy jump roughly 20-fold. The mechanism became clear in stages. A person inherits both the HLA risk variant and specific TCRA variants. An environmental trigger occurs, perhaps a flu infection, a strep infection, or another pathogen. The immune system mounts a response to that trigger. Through a process called molecular mimicry, the immune response, originally aimed at the pathogen, mistakenly also targets orexin neurons because those neurons carry sequences resembling the pathogen's own. What starts as protection becomes friendly fire. This reframed how scientists view the disease entirely. Narcolepsy is a neurological disorder that happens to involve the immune system. It's an autoimmune disorder that happens to target the nervous system. Essentially, an autoimmune attack on the hypothalamus. Research published in 2025 pushed this further. Using detailed tick cell analysis, scientists identified specific immune cells from narcolepsy patients that directly react to hypocretin-related proteins, along with expanded populations of self-reactive cells that appear to specifically target orexin neurons. These aren't theoretical. They're actual cells drawn from patients, blood showing clear signs of autoimmune activity. The genetics turned out to be more complex than a single gene. Additional genes have since been linked to narcolepsy risk, including ones involved in cell activation and survival, antigen processing, immune cell signaling, and inflammation. Narcolepsy is now understood as a polygenic condition shaped by a whole network of immune-related genes. Interestingly, when researchers examined narcolepsy cases tied to pandemics, the H1N1 vaccine associated with a spike in cases they found an association with a different gene, one regulating a factor important for neuron survival. This suggests that some people may have immune systems more prone to attack, while others may have neurons that are simply more vulnerable to that attack. Both factors appear to match more recent work from 2025, and 2026 has added another layer of differences in how genes are chemically marked and expressed in the cells of narcolepsy patients compared to healthy people. It's not just which genetic variants someone inherits, it's how those genes get switched on or off. One 2025 study proposed a 4-part framework for how narcolepsy develops genetic susceptibility, inheriting the HLA and TCRA risk variants, plus other immune-related genes. Environmental trigger exposure to a pathogen like H1N1 flu or strep immune attack on the hypothalamus, destruction or silencing of orexin neurons. Epigenetic dysregulation, broader changes in gene expression, not just in immune cells but across the nervous system. People with narcolepsy face higher rates of accidents, car crashes, workplace injuries, falls at home, along with elevated rates of depression and anxiety. Many withdraw socially out of embarrassment. Relationships. Careers and marriages can suffer under the strain, and for most of the last century, they were told it was all in their heads. Current treatments help manage symptoms, but don't address the underlying cause. Stimulant medications promote wakefulness. Other drugs affect sleep architecture and can reduce cataplexy, though often with significant side effects. Newer options. Targeting histamine receptors show promise. None of them stop or reverse the immune attack on orexin neurons. None of them cure the disease. Understanding the immune basis of narcolepsy has opened new treatment avenues. Early immune intervention to potentially prevent the disease in genetically at-risk people exposed to a triggering infection. Anti-inflammatory approaches to slow progression. Orexin replacement delivered directly to the brain. Targeted immunotherapy aimed specifically at the self-reactive cells while leaving the rest of the immune system intact. Orexin receptor agonists, which activate the brain's orexin receptors directly, rather than trying to raise orexin levels, some already in clinical trials, with early signs of better symptom control and fewer side effects. Genetic testing now also allows early risk identification. People carrying both the HLA and TCRA risk variants can be monitored more closely, and if suggestive symptoms appear, they can be tested quickly using cerebrospinal fluid orexin measurement, the gold standard diagnostic test. Earlier diagnosis means earlier treatment and better outcomes. Researchers increasingly see narcolepsy not as one single disease, but as a spectrum. Some patients have clearly documented low orexin levels, others have cataplexy without documented deficiency, and others have excessive sleepiness without cataplexy at all. The old dividing lines between type 1 and type 2 narcolepsy are blurring as the underlying diversity becomes clearer. The H1N1 pandemic created something like a natural experiment. Countries that used the pandemrix vaccine saw sharp spikes in narcolepsy, especially in Scandinavia, while countries using other H1N1 vaccines didn't see the same pattern. Genetic analysis of the pandemrix-linked cases showed a different genetic signature than classic narcolepsy cases, suggesting that specific vaccine formulations and viral strains can trigger the disease in genetically distinct groups of people. Other research directions include gut microbiome studies, exploring whether certain bacteria might prime the immune system toward attacking orexin neurons, or influence immune tolerance in ways that allow the attack to happen. Biomarker research using blood tests, protein, and metabolite profiling, and antibody cell characterization to track disease activity, potentially paving the way toward fully personalized diagnosis and treatment. Major institutions, including Stanford INSERM in France and universities and research centers across Europe and Asia, have collaborated closely on this work alongside sustained support from national health research funders. Patient advocacy groups have also played a major role helping recruit participants. for studies, pushing for research funding, and giving the human urgency that often drives scientific breakthroughs as much as data does. Future possibilities include gene editing approaches to correct disease, related genetic variants in immune cells, cell therapies to regenerate or restore orexin neuron function, and combination treatments that tackle the immune attack, restore orexin signaling, and support neuron survival all at once. Turning basic research into approved treatments typically takes a decade or more. Safety and efficacy need rigorous proof, and regulators must sign off. But the pace of discovery over the last 10 years offers real reason for optimism. Ideas that seemed unexplainable in 2010 or untreatable in 2015 are now within reach. Perhaps the biggest shift in thinking is this narcolepsy isn't fundamentally a sleep disorder. It's an immune disorder that happens to disrupt sleep. You don't treat it by managing sleep architecture. You treat it by addressing the immune attack at its root. That reframing is now shaping the next generation of therapies. For nearly a century, narcolepsy was a mystery and patients suffered while doctors had no answers. Then, piece by piece, genetic studies revealing the HLA-link genome-wide analysis, uncovering TCRA variants immune profiling, confirming self-reactive cells, epidemiology, identifying triggering infections and epigenetic research, revealing genosilencing mechanisms. The picture came together. It wasn't a straight path. Some early leads like antibodies against a protein called tribbles, too, didn't pan out. But the overall trajectory kept moving toward deeper understanding. Today, people with narcolepsy have something earlier. Generations of patients never had scientific proof that their condition is real and biological, not psychological. Their neurons were under attack by a misdirected immune response. None of it was their fault. The implications reach well beyond narcolepsy. Over 100 human diseases are linked to specific HLA variants. Why does one variant predispose someone to narcolepsy, another to celiac disease, and another to type 1 diabetes? How do these HLAT cell receptor interactions decide which tissues get targeted? Narcolepsy research is helping answer these broader questions about autoimmunity and the discovery that cell receptor genetics, not just HLA genetics, can drive autoimmune disease, has opened new research directions across the field. If immune-triggered gene silencing turns out to play a role in other autoimmune neurological diseases, conditions like multiple sclerosis or myasthenia gravis, entirely new treatment strategies could follow. For people living with narcolepsy, today, knowing the mechanism is understood offers real hope and validation, transforming what once felt like an unexplainable curse into a treatable medical condition. Clinical trials, prevention research, gene therapy, and cell therapy approaches are all moving forward, suggesting that narcolepsy care could look very different within the next decade or two. In the end, this is a story about persistence. Researchers who refused to accept that a disease affecting thousands should stay a mystery, and patients who kept searching for answers through decades of being dismissed. It's a story of how genetics, immunology, and neuroscience can come together to solve even the most puzzling medical questions. The immune system remains one of biology's most remarkable achievements, but this story is a reminder that its power can sometimes turn inward. Understanding exactly how and why that happens is what gives us the ability to intervene, treat, and ultimately heal.